Hello and welcome to Biorusage Lessons. Now that we're finishing our unit about cells, let's talk a little bit about the origin of cells. More specifically, the origin of life, very briefly, but the origin of eukaryotic cells, which are very complex, lots of stuff in there. So there is a special chapter just for that, for us to discuss this. Okay, so let's go for the origin of cells. So some people think that maybe we can make a cell like that. Ooh, just start putting things together and voila, you have a cell. But this is not how cells build each other. That's not how a cell builds other cells. Nowadays, cells come from other cells. We don't build the cells from nothing. By the end of sorry, not the end theory, by the cell theory, every cell comes from another cell. So we have a cell that divides and makes two cells, and divides and makes two cells. We cannot put there just lots of phospholipids, DNA, RNA, ribosomes and stuff, and boom, we have a cell. It doesn't work like that nowadays. Okay, so a cell can only be formed from another cell. That's the cell theory. So you have a cell, divides, and boom, we have two cells. That's the idea on how cells are formed nowadays. A dividing cell makes two cells. So cell come from cells. So we are not going to be able to do like in this diagram, just put the parts together and boom, voila, we have a cell made. Now it's not going to be working like that. Actually, cells are going to come from cells. But then comes the question, okay, where how was the first cell made? Where did the first cell came from? So, first cells were definitely not an eukaryote. Eukaryotes are very complex. They have a nucleus, they have mitochondria, they have the plasmatic reticulum, Golgi, all that stuff. So they are very, very, very complex. So certainly it wasn't an eukaryote. The first cell probably came from non-living material, and it was probably a structure. We have an idea nowadays that the RNA was probably the first self-replicating molecule. Of course, we cannot go back in time to see how it was back in then, but RNA is a very good candidate for a self-replicating molecule to do the role that today DNA does. We'll discuss this more when we discuss biochemistry of nucleic acids. If the molecules assemble together, they could make a cell. So there is a possibility that this have happened. We'll see more about that. If you are curious about how this could have happened, you can read this book called Life Ascending by Nick Lane. It shows a very good view of the origin of life. There will be a discussion later about the primordial soup, the Urey-Miller's experiment, that if we put methane and hydrogen and ammonia together with water, heat, sparkles, then we can make organic molecules and stuff. Yes, this experiment was done, but nowadays we know that things probably wasn't exactly like that. There's another idea. The origin of life today, we believe, comes from other places, especially from the bottom of the ocean. We are going to have this discussion, but yeah, not right now. Number two, a cell has been artificially created. So human beings, we have in the lab created a cell from scratch. But we built the DNA, okay, we put the sequence of bases that we wanted there, the nucleotides, to be coding for something. We got an empty membrane, but the membrane was already there, and we filled this membrane with the DNA. So it's not a natural way of doing this, but actually it was possible to do it artificially to make a cell from nothing. But today, if organic molecules try to assemble like that, it's probably going to be devoured much before it's capable of establishing itself. The other cells around are not going to get something that is very simple and very not very well tested yet to be going on into life. Okay, let's talk about evidence for the idea for life. So nowadays we know that every cell comes from dividing cells. So prokaryotes, eukaryotes, any cell comes from a dividing cell. So that's something that we know nowadays that is happening. So it doesn't put much of a light on how the first one came, but it puts on a light showing us how the ones that are around, they are around. They just come from another cell that divided, and that comes from other cells that have divided, and so on. Okay. Another discussion that we're going to be having is the idea of the spontaneous creation of life. Once upon a time, people believed that if you leave organic material, life would appear from nothing in there. So if you leave meat, maggots would just appear from nowhere. If you leave dirty clothes in a corner of your room, rats would be created. But Pasteur has shown that this is not the case. If you cover the meat with a little net and flies don't manage to lay eggs on the rotting meat, then maggots are not going to be created in there. So actually maggots are not created. Maggots do not appear from nowhere. Maggots are laid there by, when the flies, they lay eggs on the rotten meat, the maggots show up. So no, it's not spontaneous creation of life. Okay. Louis Pasteur was the guy that came up with this idea especially for microorganisms, that things don't show up from nowhere. And he made a very elegant experiment with meat broth, the juice from meat that's very easy to rotten. And if you expose this broth to the air, very soon lots of bacteria will be growing there. And people thought, okay, so then you have this life, the bacteria stuff that is growing there and appears from nowhere. But what Pasteur showed is that it's not the case. He boiled the broth in this pot over here with a swan neck. It's this twisted tube over here that do not allow air particles to travel from the outside all the way into the inside. The air particles, they get trapped in here and they don't manage to pass through this point. And the broth, after being boiled, they stood untouched. They stood sterile. They didn't rotten. Basically, the guy invented pasteurization. That's why the name comes from Pasteur. If you twist the neck and you contaminate this area over here, like this, you contaminate this area over here, and then after a while, the broth will be all rotten because the bacteria will enter the swan neck and you contaminate with microbes from the outside. So actually, he showed that the microorganisms that are coming from outside, and if you boil it and you avoid the microorganisms from entering, then it stays without life in there. This is the idea of Pasteur. Boiled broth into the swan neck tube, no contamination, but if you remove the swan neck, the curved neck, then you have bacteria growing there and making things to be all rotten. So basically this is the process of pasteurization. We heat up food, we kill the germs, we seal it, so we avoid the recontamination with life coming from outside. And with this experiment, Pasteur showed that life doesn't show spontaneously. Life comes from other life that were somewhere and then get into the broth to be growing. Okay. As we're talking about the origin of life, that's not spontaneous. No, there will be a big discussion about that in the future. But now I would like to talk to you about the origin of eukaryotic cells. Prokaryotes, the origin of prokaryotes is something very discussed, and it's a major thing in the universe. But prokaryotes are very, 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 very simple. It's quite possible for us to imagine structures organized in cells, making a membrane, and with some stuff inside and giving rays for a very, very primitive prokaryote. But eukaryotes are much, much, much more complex. We have many more structures inside, and how that complexity came to be. And then that comes the endosymbiotic theory. The endosymbiotic theory says that even if you go adding molecules and molecules and molecules and molecules for millions and millions and millions, actually billions of years, we will end up having a simple cell maybe, but the eukaryotes are way too complex, and it wouldn't be happening just by adding molecules and molecules and molecules. So how did they came up to be? So the endosymbiotic theory states the following: once upon a time, a bacteria, a big prokaryote, ate a small prokaryote. That was how things happened back in that time. Actually, it's still today. Big cells eat small cells, big microbes eat small microbes. So there was there a big prokaryote that engulfed, ate a small one. So after a while, we had a big cell that has just eaten the small one, and the small one now was surrounded by a vesicle, by a membrane inside the big cell, and the idea is that this cell would be digested. But instead of digesting, the big cell kept the small cell alive. Now this small cell was doing something that was quite interesting, was breaking organic molecules and releasing energy. The big cell said, hey, that's a nice trick, let's keep you alive in here. And the small cell kept itself doing its biochemistry wizardry, and the big one took advantage of that to be able to get energy from big molecules being broken by the small cell. And this showed up the first association of a mutual beneficial relationship, a symbiosis between these two organisms. And the big one now has a tiny little slave, and the tiny little slave actually has a big protection that go after the food, bring the organic molecules for it, and then it just have to do its tiny little part and to stay alive. With this idea, that would create organelles inside of ourselves. That would mean that some organelles that we have inside of us, they are actually not organelles, they are not created by ourselves, they come from outside and they are actually bacteria. So that's the case. If this would be the case, these little organelles, it would be expected that they would have their own DNA. So mitochondria and chloroplasts, they have their own DNA. And not only that, the DNA is circular and the DNA is naked, just like prokaryote DNA. Oh, look, so we have an organelle inside of us that has its own DNA, and the DNA is circular and naked, just like prokaryotes. Oh, so this is an evidence that these guys actually are or used to be free-living prokaryotes. Another thing is that it would be expected these organelles to have two membranes. The one that they had in the moment that they were eaten, and the one that surrounded it once they were eaten, the vesicle. And the mitochondria and chloroplasts, they both have two membranes. They have a membrane surrounding, another inner membrane, so they have a two membrane system. And yeah, this is supporting the idea that these guys once upon a time were free-living bacteria, and then they got eaten, surrounded by a vesicle, and now they are a two-membrane organelle inside of us. They should have 70S ribosomes, or at least prokaryote-like ribosomes, and mitochondria and chloroplasts they do have. It's not exactly 70S, but it is prokaryote-like ribosomes. So they have their own ribosomes, and the ribosome is different from ours. It's no ribosomes from prokaryotes. So yay! Another evidence that these guys were free-living prokaryotes. Mitochondria and chloroplasts can even live alone for a while if given the right conditions. If you give them the proper conditions that it's found inside of a cell, these guys can live alone. Of course, they are so much associated with us today that they are not capable of living free in the environment. If you put mitochondria in fresh water, it's going to be dying. But in the same way, our cells are not going to be able to live much without mitochondria anyway. So we have an association, and these guys are there living with us. And this is the evidence that we have that mitochondria and chloroplasts, actually they used to be free-living bacteria, and now they are kind of bacteria living inside of us. Okay, so we have here an image showing the idea of a chloroplast being created. So we have a kind of cyanobacterium, which is a bacteria that do photosynthesis, that has been surrounded, and by being surrounded we have a two-membrane system, and that made the double membrane chloroplasts that do the photosynthesis for our plants that we have around here. And here you have a big diagram showing an idea of the formation of the nucleus. This was not endosymbiotic, but it was with the invagination of membranes, just like mesosomes, but mesosomes folding and making that structure over there. But most importantly for the endosymbiont, here we have the mitochondria being eaten. You have a protobacteria that have been eaten, surrounded by membrane and making mitochondria. And then later you had a second event of endosymbiosis with a cyanobacterium entering. Mitochondria do cell respiration, aerobic cell respiration, and the cyanobacteria doing photosynthesis for plants. Plants have both, and we have only one of them. Okay. My suggestion for you now is to take a look on this video. It's going to be talking about stuff that you will see in biochemistry during energy part, but it will give you a glimpse on the idea of endosymbiosis. Okay, it's a very good video, not very long, it's quite short, and this will give you a more broad view on this endosymbiosis idea. Okay, after you've done that, take a look on this quiz, and I'll see you in class. Bye bye.